Hey there, we are going to go over our meiosis notes. This is packet page 81 and 82 for us, and I've actually already cut these out. So basically what I did is I took both of the meiosis notes, packet pages 81 and 82, and the second part, meiosis 2, right, I cut it out and I just glued it straight down into my notebook, right, like this. And then the other page, the meiosis 1 page, I just put a piece of tape up here, right at the spine of the notebook, and I taped it on top of that first page that I just glued down. So now I've got both pages of my notes here on the same page of my notebook. Okay, so I glued down the meiosis 2 first, and then just put a piece of tape up on the top here to create a little hinge for me. So we're going to talk about meiosis. Now let me turn my notebook sideways so I can take some notes. So meiosis is the process of making gametes. Now, gametes are special cells in our body. They are our sex cells. Right? They are used in sexual reproduction, right, which we talked about. If you are a female, you have eggs for your sex cells. If you are a male, you have sperm. Right? And this is true for any animal that reproduces sexually. You will have egg or sperm. If I'm a plant, I have we call it pollen. Right? They're still eggs, but then they have pollen instead of sperm. But it's still gametes. They're sex cells. These gametes need to have half the DNA of a normal cell, and we call this haploid, right? And that kind of sounds like half, haploid, half. So think about humans, right? Humans, we have 46 chromosomes. So at the beginning of meiosis, we start with what's called a diploid parent cell. So, right, it has a full set, right? 46 chromosomes. And at the end of meiosis, we will end with 4 haploid daughter cells, meaning they're going to have half the number of chromosomes. So, right, if I were to do some simple math, right, if I were to solve for n, right, half of 46 is 23. So we started with 46 and we're going to end with 23, right? And there's a reason our sex cells, our gametes, have to be haploid, because they're going to go through fertilization and sexual reproduction to become diploid again. So just taking kind of a quick look at meiosis, it's split up into 2 parts, right? Meiosis 1, we're still starting in interphase, right? The parent cell replicates its DNA, and we are diploid, or 2N. We go to prophase 1. The nucleus starts to break down. The homologous chromosomes pair up, right? You've got 2 copies of each chromosome in your body. So 2 copies of chromosome 1, 2 copies of chromosome 2, so on and so forth. But what happens is they kind of hug or switch some of their DNA, right? They cross over. So some of the genetic information, on this chromosome gets put onto this chromosome over here. So it's called crossing over, or kind of randomly exchanging genes. Then in metaphase, right, the spindle fibers attach to the chromosomes, right, and the homologous chromosomes line up at the middle of the cell, but they line up randomly, and we call this independent assortment, right? There's no law or rhyme or reason as to what order or what place those 46 chromosomes line up. And then, of course, we anaphase, pulling away, telophase, and cytokinesis, right? We get that pinching of the cytoplasm. But as in mitosis, right, this would have been the end. But that's not what's happening here, right? After telophase, we actually go back to prophase. So the nuclear membrane dissolves, the chromosomes are still tightly packed up, but the cells are now haploid, right? I started up here, 1, 2, 3, 4, but now I'm down to 2 chromosomes. So they're no longer homologous chromosomes, they're now just a double chromosome. So after prophase 2, metaphase 2, lining up in the middle again, anaphase 2, right? These sister chromatids get pulled apart. We're down to just that single chromosome. Telophase 2, sorry, typo. Telophase 2, that nucleus begins to form again. And then finally, cytokinesis 2, we end up with 4 haploid daughter cells. Because again, right, we started up here with 4 and we end with 2, right? And if I am a male, these will turn into sperm. If I'm a female, they will become eggs. So that's part 1 of our meiosis notes. Let's take a look at part 2. So part 2, I need packet pages 62 through 64, which I have here, and I have cut them out already. But if you want to take a minute to pause this video to cut these out, go ahead and do that. All right, so we are going to tape these together so that we can fill in our notes. So I'm going to try to tape this so I can still write and do everything. I probably grabbed too big of a piece of tape. Let me grab a little bit smaller piece of tape. There we go. All right, so lining it up. Does that look good? It's off by a little bit. Okay, right there. All right, so I'll just put in a small piece of tape right here, and then that way I can flip it over now. Use my big piece of tape to actually tape it down. There we go, much better. All right, flip it back over. Do that again for down here. Line it up. Okay, it's close enough, I guess. I'm not really sure. It moved on me. That one was a little bit too big, but we will survive. Flip it back over. Tape down on the back side of the page again. All right. Here we go. So got my meiosis notes taped together. Now I can fill them in. Perfect. So starting in interphase, right? Again, before DNA, we're in G1. So, right, this is part of the cell cycle where the cell is growing. The cell is diploid, right? Because it contains 2 chromosomes. So let's go ahead. We'll coloring this one blue, and I'm coloring this one blue, and then I'm coloring this one red, and I'm coloring this one red as well. And I guess I'll probably switch to black to right in here so we don't confuse ourselves. S phase, right after DNA replication, right? We now have homologous chromosomes, right? My cell is still diploid, but we have now replicated our DNA. Prophase 1. Oh, sorry, I forgot to color. Let me go back for a second. Yeah, color that one in. That's my red chromosome. And then my blue chromosome. There we go. Okay, so prophase 1, they have now condensed into chromosomes, right? They look a little bit fatter than they did before. There we go. There's the red ones and then the blue ones. And I'm just kind of quickly coloring them in. I don't really care all that much. But my DNA condenses into chromosomes. All right, the homologous chromosomes, they kind of are laying on top of one another, right, which causes crossing over, right, which increases genetic variation, right? It's actually one of 3 events that leads to increased genetic variation. So from here on out, we're going to be careful with how we color these because we need to show that crossing over has occurred. So down here in metaphase, right, where they're lined up in the middle, right, these solid ones used to be solidly red, but that's actually not the case anymore. A small chunk of the red chromosome has actually been crossed over onto the blue like this. So I've now had crossing over where part of my red chromosome is now on the blue chromosome and the blue chromosome is now on the red. So we're showing crossing over or exchanging of genetic information, which increases genetic variation, right? The same 2 people, female and male could have 100 offspring, but because of crossing over, right, they won't always have the exact same genes. So metaphase 1, our homologous chromosomes, right, line up in the middle. And this, again, is called independent assortment, which, again, can increase genetic variation, right? So, the fact that this big red chromosome and then this small blue chromosome ended up on the left was completely random. So there's no rule or law or anything telling the chromosomes where to line up, which means at the very end in our, 4 haploid gametes, we have no idea which chromosomes with which genes will end up in which cell. So it's completely random. So far we've had 2 events to increase genetic variation. In prophase 1, we had crossing over. Metaphase 1, we had independent assortment. I've said it 3 times now, and I'll probably say it again because it's pretty darn important. Okay, moving on to Anaphase 1, we've got our spindle fibers that will pull the homologous chromosomes away from one another, right? So this was my mostly red one. So there's red, and then this one was my mostly red one. We go, and then this had a chunk of red, this had a chunk of red, and now I can do the blue, right? Chunks of blue there, and then these were blue. So my homologous chromosomes are being pulled away from each other, right? My spindle fibers and centrioles that we learned about mitosis, right? They're still at work here. And then telophase 1 of Meiosis One, new nucleus begins to form. New nucleus begins to form. Each cell will now have one of each chromosome, so it will be haploid. So, right, this went over here. There we go. And then this one's got small red chunks on it, but this one was red. Right there, this had red chunks on it. And then my blue, right there, right there, and then this one was blue. So we're not finished, right? Even though this new nucleus started to form, it'll actually go away again because we now need to head back into prophase 2, right? Where the nucleus dissolves again. We don't have crossing over again, right? But now we just have our chromosomes, right? They're no longer homologous. It's just, right, that double chromosome. So this was my big red one right here. And this had small red pieces. And then this was my small red one. And then this had small red chunks. And then my blue, small one here. And then there's my big blue chromosome, small chunks. So, right, telophase, right, cytokinesis, that pinching of the cytoplasm to create 2 daughter cells where we continue, right, into prophase 2. So after prophase 2, we have metaphase 2, where the chromosomes line up at the middle, right? Basically, just like they did in mitosis. So, right, because we no longer have our homologous chromosomes, we're back to just our single chromosomes. And I got to make sure I keep up with my coloring. I don't want to lose track of my chromosomes. So they're lined up in the middle, and I still got to show crossing over, right? There's no independent assortment here, right? Nothing new is happening. We're just having the chromosomes line up in the middle. Anaphase 2, right? Our spindle fiber pulls the sister chromatids apart or away from each other to the opposite ends. And then I'm going to go ahead and write down telophase 2, right? A new nucleus forms. After cytokinesis, we end up with 4 haploid gametes, right? And they are not identical at all, right? So this side, you go back and draw it so we can see what's going on here. There's my red, and then here's red right there. And then the blue. So because of crossing over, all 4 of these chromosomes, or right, these 4, and then even these 4 over here, right, they are all just a little bit different because of crossing over. And then again, right here, like that. And then right here, there, and the blue one, right, like that. There. This one was my blue one. Okay. Trying to make sure I don't get confused. Red, 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 right through here. And then this is my big red one. There we go. So each of these gametes that we made are completely different from each other, right? Because we had crossing over. So right, they're all just a little bit different from each other. They maybe one got the gene, for blue eyes, one got a gene, hitchhiker's thumb, but they're all just a little bit different from each other because of that crossing over, no two gametes that are produced will be the same. In this example, this would be meiosis for a male, because we ended up with 4 sperm in the end. But again, the same thing would happen with females, except we would end up with 1 usable egg and 3 polar bodies, which basically are junk. They get thrown away. And a female only ends up with 1 actual viable egg that could go on to be fertilized. If you have more questions or you're interested in what happens with females, let me know. We can talk about it some more. But overall, right, the goal of meiosis is to create haploid gametes that will then go on in fertilization and sexual reproduction to become diploid again, right, to have a full set so that we can reproduce. So let's put this in our notebook. So what I'm going to do is I'm going to tape down this front page right here into my notebook. I'll put a piece of tape here. I'll put a piece of tape here. And then I'm just going to fold this up so that I can still see, right, the title, meiosis, and then just kind of, right, crease it down. And then I'm going to put another piece of tape here, right, so that we don't want anything hanging out of our notebook, right? We don't want it to get caught on anything and accidentally rip out and destroy our notes. So we want to make sure it's secure. So there we go. So I just put a piece of tape here at the top, 2 on the sides, 1 down here, but I can still open up my little foldable or whatever it is, and I can see all of meiosis. So there we go. That is our notes on meiosis. If you have any questions or concerns about what we talked about, please be sure to let me know.